Hi everyone, my name is Salwana Arina binti AKHIR, my metric number 23DBT20F2005. Okay, today I'm going to show you guys about SDS page. For information, the theory behind the sodium diacetyl sulfate polyacrylamide gel electrophoresis SDS page is the separates proteins based on size through the use of a tracking and resolving gel. The sample that will be obtained are loaded into the well after being combined with a buffer containing 30% glychloride SDS and toys. These are the material needed for this experiment. The first step for this experiment is increase the heat block temperature to 95 derajat Celsius. And set a micropipette to 13 UL and attach a tip and save a column. Next, draw up the well-type cell culture and close the white-type cell culture. Next, dispense the well-type cell culture into the white-type cell tube. Then, repeat the procedure to transfer 30 UL of the pancreas cell culture. Next, repeat the procedure to transfer 13 UL of the added 4 cell culture. Next, repeat the procedure to transfer 30 UL of the added 7 cell culture. Next step is set a micropipette to 10 UL and attach a tip. Then draw out 10 UL of 4X SDS sample buffer and close the 4X SDS sample buffer. Next, dispense the 4X SDS sample buffer into the white type cell tube. And repeat the procedure to dispense 10 UL of 4X SDS sample buffer into the pancreas cell tubes. Then repeat the procedure to dispense 10 UL of 4X SDS sample buffer into the edit 4 tube. And repeat the procedure to dispense 10 UL of 4X SDS sample buffer into the edit 7 tube. The protein ladder ready contains SDS sample buffer. Next step is move all of your 4 sample tubes to the heat block. And set the heat block timers to 10 minutes and press start. After that, remove all of your sample tube from the heat block. Next step is move all of your sample tubes to the microcertified and briefly spin the tube. Then remove the tube. Next step is place the polyacrylamide gel in the electrophoresis box. Then fill the gel cassette with 1 egg running buffer and remove the comb. Next, fill the outer gel electrophoresis tanks with 1 egg running buffer. After that, set a micropipette to 20 UL and attach a tip. Then draw out the protein ladder and move the micropipette to the first length of the protein gel. Next, dispense the protein ladder into the first well and dispense 20 UL of the positive control into the second well of the gel. Then, dispense 20 UL of the negative control into the third well of the gel. UL of the negative control into the third well of the gel. After that, dispense 20 UL of the added 4 cell into the fourth well of the gel. Then dispense 20 UL of the added 7 cells into the fifth well of the gel. Final step is cover the electrolysis box and connect the red and black cables to the power supply and the lid of the gel electrophoresis box. Then turn on the power supply, set the voltage to 150 voltage and set a timer to 60 minutes. Then start the power supply and wait until the time has elapsed. After that, turn off power switch and unplug the electrodes. Lastly, inspect your gel. Okay, lastly, this is the result for this experiment.